And so Laura asked me to give you some more detailed information about CASK, the differences in CASK mutations. And before I will go into the details on CASK and mutations, I thought it was better to give you some more general information about genetics and DNAs and mutations. So just start with this kind of photograph, which is actually very old, but this is how human genetics is working and functioning. And you immediately see on this photograph that we all inherit half of our genetic material from mother and half of the genetic material from father. And that is actually the basics of human genetics. So we can ask, where do my genes come from? And you immediately learn that they come from mother and father. And if we, as you can see here, if we think or if we imagine that our genetic material is like a puzzle, you see that a child inherits half of the puzzle pieces from mother and half of the puzzle pieces from father. And to go more into the genetic terms, you know, see that we all have 46 chromosomes, and you see that 33 chromosomes are inherited from father and 33 chromosomes are inherited from mother. And what you can also appreciate here is a pedigree, and this is actually what human geneticists are drawing during genetic counseling, the pedigree of your family. And you see this symbol here. This is a father or male, and here the circle, this is a female. So next, it is clear that our chromosomes are in the nucleus in all our cells, in all our bodies. And so I just give you a more detailed explanation about our DNA. And you see the chromosomes here. You can zoom into the chromosomes to see the DNA, and you see the DNA is here indicated as a blue line, and the blue line is wrapped around these green structures, and these are proteins. And on the left side here you see the DNA, the naked DNA, and our genes are made up of DNA. So here on the right, you see the DNA double helix, and we will have a closer look into the DNA. And you see here these letters, which are TGCA. And here you see the chemical names for these abbreviations: adenine, cytosine, guanine, and thymine. And we use the abbreviations for this, ACGT, and this are the letters of the DNA. And so in more technical terms, these are the so-called DNA bases. And these DNA bases can be mutated, and mutations in a certain gene, in a mutation of a certain base, can lead to disease in humans. So here you see this could be the DNA sequence. So these are a lot of bases in a row, and these kind of sequence can represent a gene. So on the next slide, I asked the question, how do genes that cause disease differ from healthy genes? And the answer is, of course, what you know, by a harmful mutation. And here I will give you an example how easy we can explain a mutation. So here you see again a piece of DNA and the letters of the DNA, the bases, AGG, CTA, and so on. And if we translate the DNA into another protein, so the DNA is actually what is encoding our proteins. But here I will not bother you with the protein. I think it is easier for you to understand that here the DNA codes for a word. So here you see the word rabbit, and everybody of you, of course, understands it. And for you, it is clear what is a rabbit. here on the right side, here's a nice picture of a rabbit. And so here we can say that this gene is healthy because the word rabbit is something what all of us are understanding and which is quite clear to you. So next, if we change one base or one letter of the DNA, so this G is exchanged by a T, and you see now this sequence is changed by a single letter, and then now we look what this causes to the word rabbit. You can immediately appreciate that now the word rabbit is mutated because you cannot anymore understand this word if we say rabbit. This makes no sense, and this is a mutation. And so we can say here that this gene is mutated, and we have also a changed rabbit. Okay, so what kind of different kind of mutations we can find in genes? And here in the upper part of this schematic, you see the normal DNA sequence, which starts with T, A, C, T, and so on. And then I will guide you through all the different kind of mutations. So in this part, which is called substitution, in this kind of mutation, there is a single base. So here, as you see in the normal sequence, the T is exchanged by a C. So this is a base change from T to C. So next is a duplication of one base, of a single base, and this you can see here, which means that this cytosine, the C, is just doubled in the DNA. So this is another cytosine in the DNA, in the gene, and this can also cause disease. Here in this row, you see an insertion of a base. So between C and T, which is a normal sequence, as you can see here above in the normal sequence, DNA sequence, you see an insertion of a G, which is guanine. And this is also a kind of mutation. Last mutation is a deletion of a single base. You see here that one base is lost, which is a thymine here. So the T is getting lost in the DNA, and this is could also be the cause for disease. So all of you have seen what kind of different mutations we have in the DNA, but of course, the consequences of mutations in DNA can be quite different when we translate DNA into protein or when we want to produce a protein. And this is also happening to CASK. So therefore, I have chosen another example to present you the consequences of mutations. Here you see in the upper row, you see ATG, TTG, and so on. So this is these are the letters of the DNA, and this is a normal DNA sequence. And if we translate this DNA sequence into a sentence that everybody of you can understand, so this is an example that DNA has all you can ask for. So this would be the normal sentence, which would make sense to all of you. And now I'm showing you some mutations. So this is a kind of mutations where you see that DNA has all you can ask for. So the U is a bit different, but it still makes sense, this kind of sentence, and everybody can understand it, although there is a single, how to say, a minor mistake in the sentence, but if we just pronounce it, everybody will understand. And so we would call this kind of mutation a silent mutation. So next mutation is the missense mutation. And the missense mutation, here the sentence would read like this: DNA has all Lu can ask for. So Lu is of course a change in the sentence, but this sentence still makes sense to all of you, hopefully. And so the missense makes that there is some sense, but the sense is not the normal sense. It is somehow a changed sense. And then I present you the nonsense mutation. And the nonsense mutation, as you can see here, leads to a stop of a premature stop of the sentence. So the sentence is not completed, it's not full length, and it is shortened, and this sentence makes no sense at all to all of us. And so this is a kind of a nonsense mutation. And last mutation is so-called frame shift. So what you can appreciate here is that when you check the letters, then you see that we have not ALL, all. We have one letter lost, and this is an L, which got lost in the sentence, in the word. And so this makes a frame shift. So all letters are now switched to the left. And so this frame shift is also a kind of severe mutation. Okay, so these are the different kind of mutations which we can see in the DNA, and these kind of mutations affect a single letter in a single gene. And although these are so small and tiny mutations, these mutations can give rise to a severe human phenotype. Okay, now I would like to remind you that our genes are located on our chromosomes, and this, I think, this should be clear to you, of course, and this is school knowledge. But I show you now here our chromosomes, which are normal chromosomes, of course, and yet you can see that we have large chromosomes, we have very small chromosomes, and what I want to tell you here is that within the 46 chromosomes we have 2 chromosomes which determine our sex and the gender. And this is for females, it's the X chromosome. We have 2 X chromosomes which are responsible that we are females. And for the males, we have a different combination. This is the X and Y combination. So you know that males and boys have only 1 X chromosome, while girls and females have 2 X chromosomes. So why this is important? It is important because the CASK gene, which is mutated in your children, is located on the X chromosome. And here, as you can see, so females and girls have 2 CASK copies, gene copies, because we have 2 X chromosomes, while in boys and males, the boys and males have only 1 CASK gene copy, because the Y chromosome does not have any CASK gene copy. It is only located on the X chromosome. Okay, so now come to CASK and the related disorders in girls and boys. And what we know so far is that in females and girls who have a CASK mutation, there's only 1 of the X chromosome affected. So 1 copy of the CASK gene on 1 X chromosome is mutated in girls and females. And in the males and boys, the single X chromosome with the single CASK gene is mutated in boys and males and give rise to a clinical phenotype. Okay, so that are the basics, what I wanted to tell you. And now Laura asked me to give you some very brief introduction in how we found the CASK gene as a disease gene, because in 2008, which is now a long time ago, we published the first paper on CASK, and that mutations in CASK cause a specific phenotype, which has later been described as MICPCH, as you all know. So this was our first patient, which we investigated for a possible mutation in CASK. And this is now the beginning of our story how we detected CASK as disease gene. So this girl is from Germany, and this girl has a typical clinical features, which we now are aware of the phenotype of MICPCH. She had a microcephaly, she had developmental delay, and when we had a look at her brain imaging, her MRI scans, you or most of you are familiar with these typical characteristical MRI findings in CASK in females with CASK mutation. You see that the cerebellum is quite small, and the pons, which is here, is also flattened and smaller than usual, and also the brain stem is hypoplastic. So this is, as we learned later, these are the typical MRI findings which we can appreciate and identify in most of the brain imaging images of females with the CASK mutation, and also of boys with the CASK mutation. So why we got in contact with these female patients? So this female patient had a larger mutation than what I have shown you before. So she had a chromosomal mutations. So this means when we investigate the chromosomes under the light microscope, you can immediately see that this girl had some chromosomal abnormality, which is not normal. And therefore, we decided to investigate this chromosomal abnormality. So here I show you the normal chromosome, and I will now introduce you in the kind of mutation this girl had on one of her X chromosomes. So you see, this is a normal chromosome. And now what has happened on one of the X chromosomes of this girl was that there were two breaks on one of her X chromosome, one break here and another break here. So two breaks. So that means that a DNA fragment out of the chromosome broke off. And so this is the part which broke off. And now this DNA fragment rotated by 180 degrees. So it was inverted now. So this is the inverted DNA part of the chromosome. And this got now inserted into the broken chromosome again. And it was somehow repaired. So this is a repaired X chromosome of the girl. And so this X chromosomal inversion, for this X chromosomal inversion, we were interested and wanted to investigate the inversion in more detail. So here is the inversion shown of the girl. So this was not the right chromosome. This was not an X chromosome, which I showed you on the previous slide. So this is the X chromosome of the girl. This is on the left, it's a normal one. And you see the different bands on the X chromosome. And on the right, you may appreciate that here on the short arm of the X chromosome, in this area, this part is inverted when you compare it to the left one, to the normal X chromosome in the girl. So here you see that she has an inversion of one of her X chromosomes. And now we wanted to study the break points of the inverted X chromosome of this girl. So here, for example, you can appreciate that we studied in more detail what has happened on one of the inversion breakpoints. And here is a zoom which shows you that in one of the inversion breakpoints, when we zoom in and look in more detail into the chromosomal DNA of this girl, we saw that in one of her inversion breakpoints, the CASK gene is located. And then when we looked deeper and deeper, we could identify that the CASK gene was broken by one of these inversion breaks in the chromosomes, in one of her chromosomes. And so this CASK gene got disrupted in 2 pieces. So one piece of the CASK gene was sitting here, and the other gene, the second part of the CASK gene was sitting here because of the inverted part on the X chromosome. So this was our first patient where we could show that the CASK gene integrity was disturbed. And so we had the idea that this mutation caused her phenotype. But of course, if you have only a single patient, this is not enough. You need to show that there are additional patients in the world which also have a CASK mutation. And this is what we indeed identified by worldwide collaborations with other human geneticists. We identified additional children with CASK mutations, and we had 5. This was the initial publication in 2008. We had 5 affected individuals comprising 4 females and 1 male. And so by this publication, we could show that CASK is a disease gene. And when CASK is mutated, either by inversion, disruption of the CASK gene, or by a single base pair exchange, this leads to a very specific phenotype, which we already described in the first paper. And we noticed the common phenotype, which is microcephaly, MRI findings of pontocerebellar hypoplasia, developmental delay, which leads to intellectual disability later. So most of the patients are not able to walk, and all other clinical features which you, of course, know. So here I would like to show you some pedigrees of families with 1 or more children carrying a CASK mutation. And you immediately, of course, can see that in most of the families, there's a single child which is affected by a CASK mutation. So if we now go into detail and we analyze the CASK gene, or we found a mutation in the CASK gene, you see that in the affected girl here in this pedigree as an example, we identified the mutation on one of the CASK gene copies here in this female patients with MICPCH. And when we investigate the parents, the father and the mother, we see that neither of the parents do carry the CASK mutation. And this is very classical and very typical that we identify the CASK mutation only in the affected patient, but not in the parents. And so we call this a de novo mutation, which is only present in a child and not in the parents. So if you go to this pedigree, for example, in this pedigree, we have healthy parents and we have 2 affected boys. They have a healthy brother and a healthy sister. And if we now go into detail and analyze the CASK gene here in these boys who have developmental delay and microcephaly, then we identified the CASK gene mutation in both boys. And then, of course, we can also check the mother, because the father gives the Y chromosome to his boys, and the mother gives the one of her X chromosomes to boys. And so when we study the mother, it is possible that in these cases, or in this situation, the CASK gene is mutated on one of her X chromosomes, although the mother is completely normal, completely healthy, and fine. And in this kind of situation, we indicate the mother with a black dot. She is healthy, so this is the phenotype, and the black dot indicates that area of a heterozygous CASK mutation, but this CASK mutation is not impairing her health. She is healthy and fine. So, and this means that the CASK mutation, which can be found in males, can be inherited from moms, but there are several cases where also mom did does not carry the mutation in CASK. And then the same situation is in boys, that the CASK mutation is de novo, so not inherited by mom or from mom, and it occurred de novo in the single male or boy in this family. Okay, so we can summarize. So CASK mutations are quite complex. They can have different effects also on the clinical picture. So if you see here this fishing hook, which is representing the normal CASK gene, we can conclude that they are very severe CASK mutations, and you see that this is a severely deformed fishing hook, and we can have more milder CASK mutations, as here as an example with this fishing hook, which is mildly deformed and is possibly partially functioning. So this is a complicated situation, I know, with the CASK gene, but this is a very simplified explanation that we have more severe mutations in girls and boys, and that we have milder mutations which mainly affect boys and can lead to a clinical picture, but the clinical picture is not so severe as seen in boys with a severe CASK mutation or in girls with a severe CASK mutation. And now I come to one of your questions, which was related to mosaicism. So one of your questions was that you have heard about mosaicism of the CASK gene mutation. So what means mosaicism? So here, because we mainly investigate DNA, which is isolated out of blood cells, and here is the principle of mosaicism shown to you. So here are a couple of blood cells, and you can see those blood cells with a red dot and with a black dot. And those cells with a red dot, they carry the CASK gene mutation. So, and in boys, these cells have a single X chromosome and the CASK gene is mutated. And in the other cells with a black dot, you see that these cells carry the normal X chromosome, where the CASK gene is normal and intact and functioning. So this means the boys with such kind of CASK mosaicism have two kind of cell populations in the blood cells, which means they have cells which carry the CASK mutation on the X chromosomes, and they have cells which are normal in terms of the X chromosome and the CASK gene. The same situation can happen in girls. So it is also possible, although I do not know whether it has been already described or published in the scientific literature, that there are girls with a CASK mutation, which have, the girl has cells with a CASK mutation, and in addition, the girl has also cells where both copies of the CASK gene are normal and not mutated. So this would be also mosaicism in girls. And what you should know is that the kind of mosaicism can be different in blood cells and other cells of the body. So, for example, if we would investigate buccal cells or skin cells in the same person, we could get the result that the mosaicism is stronger or not so strong compared to blood cells. So there's a lower kind of mosaicism in buccal or skin cells. And of course, we cannot predict how the mosaicism for the CASK gene mutation is in the brain cells and the neurons. This is unpredictable, although we know the kind of mosaicism in blood cells and other cells of the body. So with this, I would like to finish my short presentation. And of course, I'm now happy to try to answer your questions.
So the first question is, we know that there are some children either with the exact same mutation or with very similar mutations, like deletions near the end of the gene or near the beginning. However, they some are more severe, whilst others, for example, maybe are walking, crawling. Can you explain why that might be?
So this is because we are all different. You know, even if we are healthy, we are different. And so it doesn't mean if you have the same mutation that you have the same phenotype. This is the case for all genetic diseases. You can never say that because two persons have the same phenotype, they have the same mutation, they will develop the same phenotype. Because the phenotype is modified by many factors. There are also other genetic factors which are modifying the CASK phenotype, and there are also environmental factors which are also give rise to slightly different phenotypes. So this is the easiest answer, I think.
Okay, so is it possible that there is, you know how like 50% of the brain cell, approximately 50%, have the mutated X chromosome, the mutated CASK gene active, and 50% have the healthy, in females I'm talking about, have the healthy CASK gene active. Is there the likelihood that this can be a bit skewed, that, for example, in some children, it might be more 70% of the healthy CASK gene is active, whilst actually the and vice versa? Is that possible?
Sure. So that is one factor which can modify the phenotype. That's X chromosome inactivation in the brain cells is different in different children and different females with the same mutation. And this we can never estimate. So what is the proportion of brain cells having the normal X chromosome active, and what is the proportion of brain cells having the mutated X chromosome active? And because you cannot look into the brain and isolate brain cells, so that is impossible. So of course, this could be a contributing factor for giving rise to a more milder phenotype or mild phenotype, and giving rise to a more severe phenotype, yes.
Okay, thank you. Some children seem to regress, and there does not appear to be a set age that this occurs. Are you able to indicate whether regression is a symptom of the progression of the disease based on the severity of the mutation, or is it if a child has seizures, for example, and that's causing the regression?
So I don't know how many children develop regression. So at least to me, regression does not seem to be a normal or a usual clinical symptom in all children with a CASK mutation. So if you go to the scientific literature, then you will find a single report on a female which is 31 years old, and this female might have developed a regression. So the authors have described that this female had a regression at the age of 31, and she was able to walk and to stand, and at the age of 30 or 31, she was not anymore able, she could not walk, only with support, and later she was wheelchair bound. But this girl or this female, she never could speak, she did not understand, so she was really severely disabled. And so this regression affected her motor activities and not the cognitive abilities of her. And in addition, I heard about one girl in Germany, because her father contacted me, and he also told me about her, his daughter, and she also seemed to have a regression which started earlier, I think during puberty. I'm not really sure whether CASK disorder is associated with regression in all females. We do not know this so far. We have to study the clinical course during life. And so we have to be very careful to talk about regression as a uniform clinical symptom in all females or males affected by a CASK mutation. So please be careful with this kind of term, and to think that all girls and boys will develop regression.
Thank you. So is it helpful for families with children who seem to be presenting with regression? Is it helpful for them to, I don't know, maybe be part of like the natural history study we have going with Rare X, or maybe ask their doctors if there's any study, if anybody wants to study this? Is there anything kind of the families can do to help further? Because obviously it seems to be mostly there's a lack of literature, you know, in general on CASK, because obviously there's a huge spectrum. There is a lot of literature, but it doesn't cover, you know, all the spectrums. Is it, or can families do that? Like, how do families get studied?
I think I cannot help with this, because I think it is important that there are doctors worldwide who are interested in CASK and CASK disorder, and who would like to collect a series of CASK patients. And for this CASK patients, the clinical history should be known, of course, and should be carefully documented by neurologists, by pediatricians, by MRI specialists. And so this, I think, is a problem, that we need a cohort of patients with CASK mutations which are 30 or 40 years old, and then we can see how the clinical course or the clinical history is developing, and then of course we can get more information about regression. But I think this is really not an easy task, and we need the interest of neurologists and pediatricians to collect 10 or 20 patients with the CASK mutations, and which are older now or getting older, and then, you know, you can see the course. But yeah, I'm not aware of anybody in the world who is who would like to do such kind of study.
Okay, thank you. Some children pass away, more so recently, both female and male, of different ages. Can it be said that CASK can result in a reduced lifespan? And if so, what is the cause, if known? Is it just the different mutations? Are you aware which mutations may be more likely to cause an early death? If yes, can we access this information?
Yeah, so actually this is the same problem, that we do not have many informations about patients who passed away and what were the reasons why they passed away. So, and I think at the moment we do not have enough evidence to say that the lifespan is reduced in patients with the CASK mutations because of lack of information. And so it could be so epilepsy, severe epilepsy could be a problem for patients with a CASK mutation, and a severe kind of epilepsy can lead to early death, especially if the epilepsy cannot be controlled by medication. Then, of course, this is a very difficult and severe situation, and may lead to an early death. And so boys, so let's give an example. So boys with a very severe form, with a very severe CASK mutation, they usually develop epileptic encephalopathy starting from birth. So they develop it during the first weeks or months of life. And this kind of epilepsy could be so severe that they could die within the first year of life. So for girls with a CASK mutation on one of the X chromosome, I think it is more difficult. And here we do not know about the reasons why some of the females and girls passed away. And there could be completely different reasons why they died. And yeah, so I think there's not much information on this. And I do not believe that in girls, the type of CASK mutation is predicting that this girl will have a reduced lifespan. I think this will be not possible to predict.
Okay, thank you. I think that a lot of parents will be, yeah, well, not relieved, but yeah, will like have wanted to know that answer. That basically there isn't an answer. So I'm presuming it's like the same for the next questions. It is about epilepsy. Obviously, you're not an epileptologist. There's no way to predict which children will get epilepsy, I presume, or it's just not known at the moment. And I think, is it known how common epilepsy is in the literature?
Yes, so for the CASK, for girls and females with MICPCH and CASK mutation, we know that about 40% of the girls and females develop epilepsy. So usually what we have seen is that the onset of epilepsy in these girls is between birth and the age of 10 years. But of course, epilepsy can also be developed at a later stage, so later in life. So if your girl is fine until the age of 15, it is in principle possible that these girls can develop epilepsy at the age of 18 or whenever. So you can, it is unpredictable, it is not dependent on the kind of CASK mutation. And yeah, so I think that is the point.
Okay, thank you. So one parent wrote in and said their daughter has CASK, MICPCH, MICPCH, and microcephaly, but her MRI does not show pontine and cerebellar hyperplasia. Is that possible?
So we know from the literature, from the scientific literature, that there are 2 females reported with a CASK mutation and a normal MRI scan. So we checked this literature or the paper where this was reported, and we could not find any representative MRI scans which they show for these girls. And so it is not possible for me to evaluate these MRI scans in these 2 girls. And what we have noticed is that in some female patients, the pontocerebellar hypoplasia is so mild, so the cerebellum is mildly affected and shows only a small, it is tiny, it's only a little bit smaller than normal. And so it is possible that some radiologists, neuroradiologists, will interpret such kind of MRI scans as normal and not as a slightly smaller cerebellum.
Okay.
Yes, it is possible that there is no PCH or that the PCH is very mild.
Okay, but the condition would still be called MICPCH. It wouldn't be called XLID. It would still be called.
No, I don't think so. I think we should stay with MICPCH. Although the PCH is not visible, not detectable, or only very mildly.
Okay. Are there any issues that present themselves as those with CASK that aren't obvious or apparent in the younger years?
Yeah, so we already discussed it as it's epilepsy, which can be developed later in life. Then spasticity, which means that its extremities are hypertone. Yeah, and there could be some spasticity present. Then scoliosis. And what is also important to know is that it is really recommended that all girls with a CASK mutation have regular ophthalmological evaluations and also evaluations regarding hearing. So because they can develop problems with the eyes and also with the hearing later in life, and so it is really important that you should go for regular 1 time per year or something like that to the ophthalmologist, and also for hearing tests.
Okay, thank you. That's very good to know. The next question is about the well, whether it's degenerative or not. You've kind of partly answered that. It has been said that CASK is degenerative based upon the premise that brain cells are dying. There's neuronal death. Do you agree? If not, why?
Okay, this is a very difficult question. So there was one study in the scientific literature, and they report the autopsy of a brain of a deceased 2-month-old boy with a severe CASK mutation. So this is a boy with a severe mutation, and he definitely had a very severely affected brain and also developed epilepsy. So in this brain, the scientist found signs of neurodegeneration. That means that neurons get lost. This is consistent with cerebellar degeneration in male mice. So we are talking now about mice, which have a complete loss of CASK. So also in mice and in boys with a severe mutation where there's no CASK at all, which is functional, there is at least regarding this report, there are signs of degeneration. What they also studied is the morphology of cerebellar structures in the brain of the deceased boy. And they could observe some morphological abnormalities in the cerebellum of the deceased boy. Okay, so that is what they found. We have published in 2008 our paper on the CASK gene as the first disease gene, and we also investigated the brain of a boy who deceased at the age of 2 weeks. So we also have made brain pathology, and what we found is that also this cerebellum of the boy showed morphological abnormalities, but these morphological abnormalities are completely different, are different to those identified in the boy previously published. We did not investigate whether this brain of the boy had any signs of degeneration. So this was not studied by us, but due to the morphological abnormalities identified in the boy published 1 year ago and the boy we published many years ago, we see that there are differences. Yeah, so these cerebellar abnormalities are not fitting to each other, they are contradictory, so there are differences. And this means that it is really difficult to say that just from a single individual, from a single patient, that patient with a CASK mutation or in every boy with a CASK mutation. I think these are preliminary data. We have to confirm such kind of data later by studying the brain of other deceased boys with a severe CASK mutation. And so I would be very careful to say that every boy with a severe CASK mutation shows degenerative findings. Okay, so then coming to the girls. So the girls, they have all have pontocerebellar hypoplasia, and this pontocerebellar hypoplasia in the girls and boys, I'm talking about humans, is visible at birth. So already at birth, when you do an MRI scan, you see that the cerebellum is small, too small, and that the pons is too small. At least to me, this means that we have a developmental defect, that cerebellum and pons is not developing normal during embryogenesis. And we have the cerebellum is too small. It has not the full size as a normal cerebellum. And at least to me, this is definitely pointing to a developmental defect during the at birth due to MRI scans. So if you would have a degenerative process going on from birth to, I don't know, the age of 10, the age of 20 years, then you would expect in the MRI scans that the cerebellum is shrinking. It's getting smaller and smaller, and that you have an enlargement of ventricles of all these. So ventricles are filled with water, and if ventricles enlarge, they are filled with water. They are more filled with water, and then, you know, this is a kind of a degenerative process. And so 2 years ago, there was a paper published from a group from the Netherlands, and they compared MRI scans from individuals with a CASK mutation, and they compared the MRI scans with individuals which have an autosomal recessive form of pontocerebellar hypoplasia. You may know that there are different types of pontocerebellar hypoplasia, and for the autosomal recessive forms which are caused by a different gene defect, it is already known that these have a degenerative course and are progressive. And they compared the MRI scans between CASK and autosomal recessive PCH. And what they see is that in the MRI scans of the autosomal recessive individuals with the PCH, they see degenerative signs, clearly signs in the MRI which can be interpreted as degenerative processes. These signs of degenerative processes cannot be seen in the MRI scans of CASK individuals. So, and because of these findings, they conclude that we do not have a degenerative process in females and males with CASK mutation. And this is different from that in the autosomal recessive forms of PCH. Another point is that in female mice with a CASK mutation on one X chromosome, the authors from the paper from 2022, where they investigated the brain of the deceased boy with a CASK mutation, they could show that these mice have cerebellar hypoplasia at birth, and this cerebellar hypoplasia is not getting worse. So there is no cerebellar degeneration in female mice with a CASK mutation on one X chromosome, which also, to me, speaks against a degenerative process. So all in all, I think we need further studies to discuss the point whether there are some degenerative processes present in patients with CASK mutations. But if you, I mean, I would be very careful to say that at the moment, at this point, from the scientific literature, we have enough evidence to say that this CASK disorder is a neurodegeneration. I think we need more scientific evidence from humans, from mice, especially from humans, to have to state that we have neurodegeneration.
Laura, you're on mute.
Sorry. Right, so we're getting to the end of the questions. This is about variants of unknown significance. So one mother wrote in and said, the neurologist thinks it's highly likely my son's challenges are a result of the mutation based on symptoms noted in literature and in our community group, but not knowing 100% is frustrating at times. Is there additional genetic research and information available that can help further define this?
Okay, yeah, I know. Variants of unknown significance are how to say, they make parents unlucky. And I can absolutely understand this. This is a situation which is not black and white, which means that we are in the middle of nowhere. And at the moment, we cannot say whether this variant or mutation is the cause of the disease of the child or not. It's unknown. So the only thing what is possible is that this kind of variant is included into a scientific research project, and within the scientific research project, this variant can be studied and can be investigated in terms of pathogenicity or not. Otherwise, what you can also do is to wait. And it is possible that that after 4 or 5 years, you go back to your human geneticist, and then you ask them, please re-evaluate this variant of unknown significance and try to answer now whether this is pathogenic or not, based on more genetic information what is available worldwide and about more patients with a CASK variant worldwide. And it is possible that after 4, 5, 6 years, your human geneticist will reclassify the variant of unknown significance as a pathogenic variant, so clearly being the cause of the disease in your son. Or on the other side, it is possible that they will say, no, we have now learned more about CASK and CASK variants, and we now think that this variant of unknown significance can be reclassified as a benign variant, which is not causing the disease of your son.
Okay, thank you. I'm sure she'll appreciate that. So the last few questions is about treatment options, which I know is not your area. So thank you for trying to answer any of these. So are you aware of attempts to reactivate the silence gene on the X chromosome in other diseases such as Rett syndrome and CDKL5? And if so, do you think this is something that could benefit CASK?
So as you mentioned, Laura, this is not my specialty, or I'm not, a specialist in this kind of research. So what I can say is that yes, I think it is, in principle, I think it is a very elegant or nice idea to reactivate the silent X chromosome and to reactivate CASK on the normal X chromosome and try to get it expressed. But you have to be aware that we will start as a basic scientist. They will start from the beginning. So this means they will cultivate cells in the laboratory which have nothing to do with patients. This is a cell line which they can cultivate, and in this cell line they can try to activate the normal CASK gene copy and to try to activate the CASK gene copy and express the normal protein. But when you have done it in a cell line, there are many steps which you would need to do this in a human, in the body of a human patient. So this is just to inform you, or to give you some how to say, advice that it will take years until you will have a treatment, a possible treatment for patients.
Okay, yeah, thank you. Yeah, of course. And another quick question is, I think this is more for you as a scientist, just in general, being in the area of research. Is it funding that might be restricting CASK ever getting treated, or is it scientific interest? If you could give us one piece of advice, how to, eventually get, to the end of that pathway and end up with a treatment, what do you think it is?
I think this is the most difficult question in your list of questions, and I really have no very good answer to it. I think there are options of treatments for your children, and I think this is the first point. I think we should do everything to increase the quality of life for your children. But I think, I mean, I have to be honest, and because I'm honest, I have to tell you what I have told you before, that I believe that the CASK disorder is a developmental disorder, which means that due to the fact that the cerebellum and the pons in the brain is not developing normal, and you have a developmental defect in the brain, which means the structure of the brain is not normal, you will not be able to repair this in your children, even if you find something for treatment, because you cannot reverse a developmental defect. You can try to treat children to make their life easier, I think. But yeah, I think there are treatment options, but still you have to think about the fact, or appreciate the fact, that the brain development was not normal during embryogenesis in your children, and you cannot repair this defect later in life. I mean, gene therapy is in principle, should be given a trial. So I think your children will definitely improve when you give back the gene, the normal gene. But I mean, there is a difference between treatment and cure. And so I would think that treatment is possible, but to cure MICPCH, I think this is rather impossible, because you have to imagine when is a child diagnosed with MICPCH? So normally nowadays within the first year of life, I would say, right? Or maybe second year of life. So, but within this time, you already have the developmental defect in the brain, so abnormality. You have also the brain, the neurons are developing, and in the brain of the child. And so you have already some defects in the brain, which cannot be reversed, possibly. I don't know, but you know, this is maybe different to Angelman, or I don't know to what, but maybe I'm also too pessimistic. And I think it is really worth to try to find some treatments for CASK. Even if you could, for example, diminish or reduce epilepsy, or, I mean, this would help so many parents if epilepsy would regress or disappear completely. And then I think also the child can develop better, because it is not disturbed by this epileptic seizures.
Okay, and when you use the word treat, find something to treat, you're not ruling out gene therapies. You're not ruling out that gene therapies could be the thing that could treat it. It's just they're not, a gene therapy isn't suddenly going to create like a 12-year-old, you know, a healthy, normal 12-year-old who'd previously, you know, been severely disabled. Yeah.
This is what I meant.
Yeah.
So that you try to improve, of course, the clinical symptoms of the child, especially if the child is, I don't know, 10 years or 12 years old or something like this, because I mean, it is clear to all of us, the earlier you can treat a child, the better is the outcome.
Yeah.
So I think this depends on how early the genetic diagnosis is identified, and how early you see, okay, this child has a CASK mutation and we have a treatment option. And we have treatments, we have gene therapy for other diseases, we have treatment options, but you know, most of the children improve, but they are not getting completely normal.
Yeah, no, I think that's a very important message actually to give to the parents. We wouldn't ever want to give false hope. You know, I'm also of the pessimistic viewpoint, you know, that I know my daughter's probably not going to be impacted by any treatments, and she's so severe. I mean, it'd be amazing, but I recognize, you know, even if we replaced every single CASK gene in her body, she's not suddenly going to be running around like a typical 5 or 6 year old. So, but yes, I think you've really hit the nail on the head, that for us as parents, even what other people might see as really minor improvements, for example, you know, they still be severely disabled, but maybe not have that epilepsy, you know, that for us is a real, you know, is important. And I think we need to be reaching for that. Like any improvements, we always celebrate in our community, however small. So yeah, I think that's quite a nice thing to end on. Thank you.
Yeah, I think Laura, this is the main point, and you should have the hope that this treatment will be found someday, and this is really to improve health of your children and to make their life easier and maybe a little bit more comfortable.
Yeah, thank you. Yeah, brilliant.